Hello everyone, today I'm going to show you how to do a dihybrid square in the classical fashion. The way we begin this problem is we begin to read through it just like any other monohybrid problem we've done before this. So as we read, we see that black hair is dominant to blonde hair and that being tall is dominant to being short. So let's assign some symbolism here. Alright, so we're going to now say that black hair, one big B, doesn't matter what the second allele is, you will have black hair. The only way then to have blonde hair then is to be two lowercase b's. Being tall is dominant to being short. So those are our symbolism that we're going to use. If a woman is heterozygous for black hair and being tall, so that means her genotype is big B, big T, little t. She is going to be crossed with a man who is homozygous for black hair and being short. If he's homozygous for black hair, that's the dominant trait, so he's homozygous dominant. If he's short, the only way to be short is the homozygous recessive version for that second gene. If we finish out our problem now, let's see what it wants us to actually answer. What is the probability, so percent or fraction, that they will have a child who has black hair and will also be short? So I'm looking at the possibility that this parent and this parent are going to produce a child that is this genotype. That would give them black hair. It doesn't matter what the second allele is, could be dominant or recessive for the B gene, and fully recessive for the T gene to be short. That's what we're looking for our answer. So to do it in the classical sense, I'm going to need to take these two genotypes, which each have now two genes at a time, and FOIL them to figure out what the possible gametes could be. So what that means is I got to take one letter at a time in each gene. So for example, if I start with the big B right here, if that's donated to an egg cell for the female, that could pair up with big T or little t. You're not really sure. So that means you have two possibilities to start with. But what if for that first gene, for hair color, use green, it's the little b that's given? Well, maybe that pairs up with big T, or that pairs up with little t. So now I actually have two other options I got to take into account that female could produce as an egg cell. So what I'm going to do with those four possibilities, I'm going to come up to the Punnett square and I'm going to put them across the top of this Punnett square. I could have put her along the side, on the left-hand side if I wanted to, but I'm just choosing to put her here on the top of the Punnett square. Now, for the male, if you look, you can hopefully notice already that he should be a little bit easier. He's homozygous dominant for the first trait and homozygous recessive for the second. Technically, you're still supposed to FOIL. So, for example, wrong button. Does this big B pair up with this little t or this little t? Or maybe it was the second big B that was donated. Maybe that pairs up with the first little t or the first little t. So really what you end up getting is you end up getting four sperm cells that would really all be exactly the same. So that makes our lives a little bit easier, which I really only have to write up here in the Punnett square the one possibility. Why am I going to write it another 3 times? Because each row across is going to be exactly the same thing. So it's just going to waste time and energy. So in this Punnett square, really all I have to do is do one row across and figure out 4 possible child outcomes. So when we fill in the Punnett square, we're going to keep the same letters together for the specific gene, and then we still keep dominant alleles in front of recessive alleles. So what I mean by that is this. So in the first box, I have a big B with a big B, and then for the T gene, I'm going to put the dominant allele first and the recessive allele second. And then I'm going to have in this box big B, little t, big B, big T, little t, and then big B, little t, little t. So those are my 4 possible outcomes. But what we want to find out, though, is how many of them fit this description that are going to be children with black hair and are short? So when I look at the Punnett square, there are 1, 2 that fit that description, that have at least one big B and are fully recessive for the T gene. So that means our answer out of 4 possible child offspring is going to be 2 out of 4, or 1/2, or 50%. That's our answer, and we're done. But again, like in my last video, what if the problem also asked for a genotypic or phenotypic ratio? Well, genotypic is a little bit longer than we're used to, because now if you look at the Punnett square, there's actually 4 unique variations. All right, so I would write that like this. I would do 1, who is homozygous dominant and heterozygous for the second gene, colon, 1 that is big B, little t, colon, 1 heterozygous, to 1 that is heterozygous, homozygous, recessive. You actually have 4 possibilities, so you have a 1 to 1 to 1 to 1 genotypic ratio. For phenotypic, 2 of those 4 are going to be black hair and tall, and that would be these 2 right here, this one and this one. So what I'm going to do for phenotypic, I would just write 2 black / tall, little slash there, tall, colon, to the ones highlighted in blue, they're going to be 2 that'll be black haired and short. And there's our answer. All right, let me do one more example with you guys for the long version variety of what we do for Punnett squares here. So if I drag this down and look at this example real fast. Again, round peas are dominant to wrinkled peas, and yellow peas are dominant to green peas. So let's give some symbolism. Round is dominant to wrinkled, so the only way to be wrinkled would be 2 lowercase r's. Yellow, 1 big Y I'll choose, doesn't matter what the second allele is dominant to yellow peas, which are going to be homozygous recessive. Fix that right there so no one gets confused. 2 little r's. Let's read through the rest of our problem. A homozygous round yellow pea plant. So let's figure out what that genotype is. If you're homozygous for round, you are big R, and you're homozygous for yellow, so you're big Y. That is the first parent. They're going to be crossed with a heterozygous one for both shape, and then homozygous recessive for color. So this other parent is heterozygous for shape, so that means they are round, but they're heterozygous, and they're homozygous recessive for color, so they're green, 2 little y's. What's the problem want us to answer? What is the probability that they will produce offspring with round and yellow peas? So again, a percent or fraction that they're going to produce a child who is round. So I will write this below. That means they at least have to have 1 big R. And they will have yellow peas, so that's at least 1 big Y. Doesn't matter what the second allele is. That's what we're looking for when these 2 parents are crossed. So what we got to go ahead and do is FOIL them. So again, as you can see here, both genes are homozygous dominant for this first parent, which means they can only ever give a big R and only can ever give a big Y. So they would produce 4 possibilities. I only have to write that once. I'm going to choose to write that once over here, draw my line straight down because it gets repetitive. The other parent has 2 allele possibilities for the first gene and just 1 for the second. So that means really unique options they have is big R with little y or little r with little y. That's their 2 possibilities. All right, you could say big R, little y, big R, little y, little r, little y, little r, little y, but why are you going to do that? Because you can see that really there's only 2 unique possibilities. So if I go and put them now for that parent over here, and I fill in my Punnett squares. These are all going to be repetitive, so I really only have 2 boxes to fill out. I have big R, big Y, little y, with big R, big Y, little y. Again, what we are looking for is this. Are those children fitting this description of being round and yellow? This one does, has at least one big R, big Y. This one also does, big R, big Y. So our answer for probability is, again, 2 out of 2. Or obviously, if I work that out to a percentage, really it's 100% chance every single time that I produce an offspring. Genotypic ratio is much simpler this time around. I really only have 2 options based on how I figured out this Punnett square. I have one who is homozygous dominant for being round and heterozygous for being yellow, to one option that is heterozygous for being round and heterozygous for being yellow. So this is already the reduced version of this answer. Now, if you would have went and filled out every Punnett square in this whole entire 16-box possibility, you would have had a lot more big R's, big Y's, little y's, etc., but your ratio would have reduced to exactly the same. So you saved yourself a ton of time. Phenotypic ratio, each one of my 2 possible genotypes, they're both round and yellow. So I could write it as 2 round / yellow, colon, 0. There's 0 other possibilities, or I could have reduced it to just 1 round and yellow to 0 possibilities. All right, this is the longer way to perform Punnett squares. I'm going to produce a second video for you to show you how you could do it in a little bit more simplistic fashion.